Thanks for coming out, guys. We are the Chromalia team, and we're here to talk to you today about our technology, which is we call Chromapearls, which is Mesoporous Silica for a novel human hair dye. As for any of you guys who use shampoo, conditioner, nail polish, moisturizer, or hair dye, cosmetic technology is a part of your everyday life. So companies like L'Oreal and Procter & Gamble spend millions of dollars in research every year. But I'm sure we can all think of a few products that cosmetic companies make that their products can't actually fill. So basically what we set out to do with our project is to be able to make a dye that can compete with the products that are being put out by these mega giants that would be more cheap, more safe, and cause less damage to your hair. Yeah, there you go. So the hair dyeing market in the US is $7 billion alone, and that's largely due to the normalization of hair dye usage over the past 50 years. So in 1950, only 7% of women dyed their hair regularly, compared to 75% of women today. Yeah. And it's not just fashion-conscious women who are dyeing their hair anymore. A lot of people who are doing it is now to stay away from the gray longer. So 11% of men 50 to 64 are dyeing their hair regularly, and a lot of that is to be able to be competitive in aging market, or aging when people are aging in the market or workplace. Sorry. So when you go to the store, you basically have two options of the types of dyes that you can use. There's oxidative dyes and direct dyes. So oxidative dyes are permanent dyes that cause a chemical change to your hair. So they use hydrogen peroxide and ammonium chloride, and they actually separate the cuticles, which are sort of the outer scales on your hair, and allow dye precursors to enter into the hair shaft and cause a chemical change. Whereas direct dyes are actually just physically attaching to the outside of your hair and are not as long lasting. So 80% of market share is actually for oxidative dyes. And that's because most people want a permanent color change. But the biggest problem with oxidative dyes is that they're very damaging. So when you're being able to cause this permanent change in color to your hair, it's actually stripping a layer of fatty acids from the outside of your hair that makes your hair hydrophilic and a lot more prone to damage. So this is a reason why a lot of people stop using oxidative dyes, is because over time the hair gets worse in quality until you have to stop using it. So basically, we want to be able to provide customers with a better way to get permanent color. So from the cosmetic supplier's perspective, a dye templating system would actually make a big difference in being able to develop new dyes. So a cosmetic supplier or a company might spend thousands of hours in research trying to formulate a new oxidative dye compound that would get people the colors that they want. So instead, if you could use a templating system where you could easily change the dye, you would be able to produce a whole line of products without having to put in too much research. Another thing you want to do is be able to enable repeated use. So the problem with oxidative dyes is over using it too often, you're going to cause repeated damage to your hair. So being able to enable repeated use by using a less damaging dye is highly preferable. So thankfully in the case of our project, the needs of both the cosmetic suppliers and the users are very synergetic. So what is the Chromalia solution? So we could have tackled this problem from two different ways. We could try to make direct dyes more permanent, or try to make oxidative dyes less damaging. And we actually went with trying to make direct dyes more permanent, as to be able to change the way that oxidative dyes work, we'd have to remove the chemical compounds that actually create the color change. So that would probably not work in terms of being able to create the color change. So what we came up with is our Chromapearls technology. And now Ricky's going to tell you guys a little bit more about how it works.
Thanks, Savannah. So our solution, Chromapearls, is actually a dye templating system using Mesoporous Silica as a vector to carry the dye compounds. Now, together with the particles, the dye particle compounds can diffuse into the hair cuticles and get stuck there, imparting color to it. Now, next slide. So how does our technology work? So Chromapearls is actually a level 1 dye or what's called a direct dye. So instead of chemical change, like in oxidative hair dyes, we impart color by physical attachment. So Chromapearls is engineered to be around the 203 nanometer size range so that it can be small enough to diffuse in between the hair cuticles while still be big enough to stay there due to its increased surface area and increased electrostatic interactions. So the other advantage of having silica as a carrier for the dye is that, as Savannah mentioned, as a cosmetic supplier, you want to be able to create a variety of dyes without investing too much in R&D every time you want to make a new dye. So by having a carrier vessel, instead of developing a whole new compound to change colors, what we can do is we can choose a new dye and just put it in the silica without changing too much of the manufacturing process. So why did we choose silicon nanoparticles? Well, for one, silicon is very cheap and easy to synthesize. In fact, our process only takes 2 steps, so it's a modification of the Stober process. And it doesn't require any expensive or exotic chemicals to produce. 2, silica has a very well understood chemistry. Its properties are very well defined since it's been around for a very long time, which means that any any parameters that we change will give a very predictable result. So we can tune the morphology of our silica particles to suit our needs very easily. Third, and probably most important, because we are using it for human use in the cosmetics industry, we want to be able to ensure that it's safe. And conveniently, silica is biocompatible. As you may or may not know, silica has already been used in the pharmaceutical industry as a drug vector that you can inject in your body to facilitate extended release of a drug compound. So when we first started this project, we surveyed the current box dye hair market, and we came up with a list of custom requirements that we would need our product to meet in order to sell well on the market. Now, obviously, due to the time and budget constraints of our project, we won't be able to have a product that is market ready. So instead, we took the initial custom requirements and we distilled it down to simple requirements so that we can have a prototype that would be able to demonstrate our proof of concept. So these are on the right side, you'll see what those requirements are and how we can validate it.
Okay. So this is sort of a brief outline of the different steps that we use to make our prototype so we could actually reach those goals that Ricky just set out for us. So the first thing that we actually had to do was to synthesize the particles. So we were going for that 200 to 300 nanometer range. The next thing that we wanted to do was be able to attach the dye to the silicon nanoparticles. Part of our idea there was we needed to figure out the best dye attachment method that would serve our needs. The thing that we did, the next sort of step for us was to attach the dyed silicon nanoparticles to hair, which would include determining the best application method, as well as just ensuring that we were able to create that color change so that our prototype would be valid as a commercial hair dye. So this is sort of a procedural overview of some of the steps that we made to make our silica particles. It's basically a sol gel process, and it is done in an oil and water emulsion. So basically we have the hydrolysis of our silica precursor, which is the TEOS, in water at pH 2 with a surfactant. And then we use a catalyst, which is sodium fluoride, to condensate our particles out of solution. So this process is perfect for what we're trying to do because we have a lot of control over the different parameters of our particles. So we could control the particle size by changing the ratio of silica precursor and catalyst, and we could also control the pore size by changing the amount of surfactant that we added. So these were the 2 options that we considered when determining how to attach dye to our particles. So the first thing that we considered was a physical absorption method. We could basically have the dye in a solvent and evaporate the solvent off, and then we'd just have dye just sort of physically impregnated in the particles, or into the pores, sorry. But basically, the biggest problem with that is dye retention becomes a difficulty. So over repeated washings, even when preparing the dyed particles, we noted that there was a loss of color. So this isn't very good if you're trying to create a long-lasting hair dye. So a decision that we decided to make was to try using a chemical crosslinker instead. So we used something called a silane crosslinker, and the image there is of glymo, which is what we used. And it basically can make a chemical bond between the inorganic phase, which is the silica precursors, and the organic phase, which is the dye. So it's a little bit more effective, but it also increases the process complexity. So it is another chemical that we needed to use. But overall, it served our purpose a lot better of making sort of a long-lasting hair dye. So just as a little bit of trivia, the dye that we actually used in our formulation is a food coloring that you can find in a lot of common products, including Kool-Aid, Twizzlers, and cinnamon hearts. It's called Allura Red. The reason why we used it is because it's non-toxic, it's very cheap, it's very widely available, and it also has the sulfonate groups that we need to interact with the chemical crosslinker. So while it might not be the most attractive hair color, it serves a pretty good proof of concept for our project. So this is the procedure that we use to crosslink the dye to our particles. So we added our chemical crosslinker to the dye in an aqueous solution under nitrogen purge. This is done because the glymo is very reactive with oxygen, and that can be quite dangerous. So then we add that solution to our sol gel during the synthesis process, and we leave that for 12 hours at room temperature. The reason for this is that we can actually attach the dye not only on the surface of the particle, but also in the mesoporous structure. So that means we can try to attach as much dye as possible in each particle. And then we wash the particles out, and then we get this pellet of pink particles at the end of the day. So this is actually the chemical equation for basically what's going on. So first we have the nucleophilic substitution between the Allura Red dye, the sulfonate group, and the epoxide group of the glymo crosslinker. And then we have the silane group undergoing a condensation reaction with the hydrolyzed silica precursor.
So once we have our particles, the next logical step, because we're making a hair dye, is to dye our hair. So what we did was we took our particles in solution, because once we calcinate the particles, it might experience an irreversible agglomeration, which is very hard to separate even after sonication. So we decided to keep it in solution for easy processing. So what we did was we took our solution and we submerged a sample of human hair, which we got online. It's actual human hair. And we submerged it for about an hour. And then we took it out and we washed off the particles, excess particles that were superficially attached on the hair. So here you can see the results. We actually tested it in both bleached and unbleached hair. And pictured here is the bleached hair. And this is the unbleached hair color before we bleached it. And then this is the dye bleached hair after we dyed it. So I'm going to pass around a sample of the bleached hair, or sorry, unbleached hair. You guys can just pass it on and take a look. So as you can see, we've managed to make a noticeable color change. Now, if you notice, the dyed hair is not as vivid as the original dye color, but it serves as a proof of concept that our silica particles can indeed dye hair. So next thing we wanted to find out after we've successfully dyed hair is how long do the colors stay? So what we did was we took our dyed hair samples and we washed it in a solution of commercially available shampoo. It was actually PC branded shampoo that you can get at Zehrs. And we also used regular water and we mechanically washed it using our hands, just like how you would normally wash your own hair. And the result was after 5 washing, we found that there were no leaching of the color or color change that's observable. So we actually wanted to wash the hair until the hair color came off, but unfortunately we did not have enough time. So what we did was we took, because 5 washes is in the range of current semi-permanent dyes, we think that as a proof of concept, this is enough to show that our dye can compete with existing products on the market. So we also used dynamic light scattering to validate our particle size of about 300 nanometers. So as you can see here, the particle distribution is actually very nice. It's centered around 250, but the mean diameter of our particles were found to be 300 meters. Nanometers, not meters. So to round off our battery of characterization tests, we also took a picture of the surface of the hair before and after dyeing. So this is actually unbleached hair without dye, and on the right side we have unbleached hair after dyeing. So the hair that you just saw right now. So as you can see on the left side, the hair surface is very smooth, indicating that it's a very healthy specimen of hair. Because if it was unhealthy, it would have increased cuticle raising. So on the right side, we can physically confirm that the particles have diffused on the hair surface, which causes the rough surface which you observe in this picture.
Okay, so after we were able to dye the hair, the next thing that we wanted to do to sort of further it as a to-market product is to put it in some sort of application solution that you could sell on the shelves to customers. So basically, when it comes to using an oxidative dye, usually you have the bleach compound in it, and you have to mix two different chemicals together to activate the reaction. But in our case, there's no chemical activation required, so we have a little bit more leeway in the type of solution that we could use to deliver the dye to the hair. So we actually used a cream that would sort of function just sort of like a moisturizing conditioner or something like that, that you can apply to your hair, and it has to be just thick enough so it'll keep the hair on for the duration of the testing. So these are sort of the ingredients that we use, and there's a picture of the cream and the particles embedded in the cream that we've made. So it's actually just an oil and water emulsion like most lotions, and it's 70% by weight in water phase, and the silica particles are actually in the water phase. So we basically just did some testing and changing of the different ratios of things in the oil phase to try to get the consistency right. So this is a cost analysis of what we used. The total material cost for our project was only $724. So if we were actually to do this at a commercial scale, we would be able to see the cost be lowered completely, like probably underneath around $2.
So in review, in a few short months, we've managed to make a prototype that successfully demonstrates a proof of concept. So our Chromapearls successfully demonstrated that we can achieve an observable color in both bleached and unbleached hair without the use of any harmful chemicals such as peroxides or ammonia. So where do we go from here? So because part of our solution is the template system, we want to be able to produce Chromapearls with different colors and different dye compounds that the hair industry have never even considered before because it wasn't possible with their current system. The second thing that we want to do is explore the use of our particles' mesoporosity. So what we could do, for example, is insert particles or chemical compounds that will nourish the hair, such as vitamins and conditioners, so that when you dye your hair with our product, instead of just a color change, what you could get instead is an extended release of conditioners and vitamins that will actually add value to our product. We would like to thank the following people for contributing to our project, and thank you for listening. We would like to open the floor for questions. Yes?
It's a very nice presentation. I have one question. Can you go to your SEM image?
Yes.
Do you have a high resolution?
We use the undergrad SEM.
But it still can should be high, because from here, it's hard to tell what exactly.
This is the image that we got from Howard.
Yeah, so basically we booked I think an hour and a half of SEM time that we got to have. So in the time that we had, this was probably about as high res as we could get. But sort of the most important part is we can really see the surface difference. As it's a little difficult to see exactly where the particles were able to diffuse into the hair. But you can see even on the initial SEM of the hair, we can't actually see the cuticle scales, just due to the low resolution.
So what do you think exactly looks like your particles stay on the surface of your hair after the dyeing? What do you think?
Well, we think that it might actually embed under the cuticles, causing the cuticles to raise up a bit, which is why it causes the surface roughness.
Yeah. I think if you were to take a cross-section of the hair, you'd be able to see probably, depending on how you cut it, a cuticle layer. I think there's about 3 or 4 different cuticle layers generally to a strand of hair, and you'd probably see the particles embedded within the first cuticle layer. Yeah, exactly.
Further work,
Okay, yeah, thank you.
our investor pitch.
Any other questions?
Did you try to dye the hair, just use that Allura dye, without anything?
Yes, we did.
No color residue at all?
Yeah, and basically how it works is that if you were to dye your hair with Kool-Aid or something like that, you would see a color change. So we did see that some dye would stay on the hair, but the whole thing is that if we washed that hair 5 times or so, and by the 5th wash, it's almost all gone. So basically the way that it works is that almost anything can work as a temporary dye. The whole point is getting it to that semi-permanent or demi-permanent amount of time that sort of makes it valuable. So
Using Kool-Aid is kind of like painting your lawn a different color.
Yeah.
So it might work for a short period of time, but once it rains or once you actually wash, or even condensation from the dew, will wash it away. So that's an analogy that we like to use.
any other questions?
Yes, Professor Simone.
Can you go to that slide where you have the surface of the particle and the dye and the molecule? The reaction.
This one, yes.
This cross-linker,
Yes.
is this FDA approved?
No, it is not.
no.
So the
But
reason why we're not concerned is because it's not in active form anymore. So this is actually during our reaction. This is the what do you call that? The least amount. So
It has the lowest concentration.
yeah, it's a limiting reagent. There you go. So once it's bound, it's no longer active, which means that.
You think you achieved 100% conversion?
I think so. That's what, basically when we were sort of starting this project, we were working with Pruzet in the project, and we said, after we have these particles and we have them in our cream, is it safe for us to be able to touch it?
If I am an investor and I want to ask an evidence of your conversion, how you would provide that evidence?
We probably have to do, I guess, some spectral analysis of the resulting powders to ensure that that is not there anymore. So Raman or FTIR, I suppose.
These chemistries that happen with surfaces,
Uh-huh.
it's difficult to get 100% yield.
Mm-hmm.
And also to control, did you do this in 2 steps or did you do this in 1 step?
In 2 steps.
Well, you mean the synthesis process?
Yeah.
Well, the synthesis is a 2-step process, but when we basically mix the crosslinker with the dye first, and then we add that to the sol gel.
Because in that oxide group can react with other things as well.
Mm-hmm. Yeah, so I mean, the idea is that we add the glymo and the Allura red first in solution.
You try to wash your Mesoporous Silicas to see how much of the dye is actually bonded there, did you?
Oh yeah, we did. We centrifuged and washed the
Yeah, we washed
dye.
it. That's that.
Yeah, so when we actually first did, if we were to show you a picture of what the centrifuge tube looked like.
How did you wash? Did you put it like a cake and you put water through?
Yep. And then we washed it, and then we would centrifuge it and take off the...
Is the dye soluble in water?
Yes. So if the dye would leach out, you would be able to see. It's basically the color of Kool-Aid.
Yeah, and basically in our first tests, we would actually have no dye attachment when we were trying to use it with physical absorption. So once we would wash the particles enough time, it would just be a white powder again. But this way we were actually able to get the dye to be permanently attached.
But I mean, it is a kinetic process. Over time, you would sort of detach, and some of it would end in the water. Do you know what's the half-life, of that dye, the bound one?
I'm not sure, but one thing that we did do is we did calcinate the particles after we had dyed them. So if we just had the dye as a powder with the mesoporous particles just in sort of a cuvette or crucible, and we put it in the oven at 600 degrees for 12 hours like we did for the calcinating step, it would be white afterwards because all the dye would burn off. Yeah, but we did actually calcinate it with the dye, and it doesn't stay red, but it does continue to retain color, which makes us pretty confident that the dye is pretty firmly attached onto the surface.
Right. And so this is a sort of proof of principle dye. If you look at the dyes that are relevant for, say, human hair dyeing or something along those lines, are the same functional groups involved, and are you confident that you could use the same mechanism to do the attachment?
Yeah, exactly. I mean, the thing that really holds back conventional dyes now is that they have to work with the oxidative process, and that's not something that we're not using dye precursors that are used that need to be oxidated to change the color. So in that case, we actually have a lot more of a wide range of dyes that we're able to use. So we could use textiles. I mean, the reason why we use food coloring is because it's a way of actually using food coloring as a permanent hair dye. We can do any color that we want.
You just mentioned textile. Have you somehow ventured into it?
No, but one idea that we have in textile is we can possibly use this to dye, let's say cotton, and then the runoff we could just centrifuge and the particles would settle, and we can recycle those particles and just dump the runoff water.
In terms of efficiency, that it works very well, so you don't actually have as much sort of environmental impact by being able to recycle the dye that you don't use. So that's another.
Focus on our target market, just hair.
Yeah. Sorry.
Okay. Yeah.
Is that good?
Sorry.
Oh, no worries.
Okay.
Thanks so much, guys.
Thanks.